Hello, and welcome to MDIC's Annual Public Forum. During today's session, representatives from FDA's Center for Devices and Radiological Health and Medtronic will discuss the External Evidence Methods Framework document that is being developed through MDIC for leveraging external data sources in pre-market medical device regulatory submissions. Please make sure to join in the conversation on Twitter by following at MDIC Annual Forum and using the hashtag MDICAPF2020. Now, please welcome session moderator Vandana Bhatti.
Hello, and welcome everyone to Medical Device Innovation Consortium Annual Public Forum session on external evidence methods for regulatory decision making. I am Vandana Bhatia, Assistant Director in the Division of Biostatistics at the Center for Devices and Radiological Health at FDA. I'm pleased to introduce today's esteemed panel of speakers. We have with us Dr. Daniel Kanyos, Director of Office of Clinical Evidence and Analysis at CDRH at FDA, Dr. Ram Tiwari, Director of Division of Biostatistics at CDRH at FDA, and Dr. Ted Listick, Senior Director, Corporate Biostatistics at Medtronic. Please feel free to type in your questions in the Q&A box at the bottom. With that, I will now turn over to Dr. Kanyos.
Hi, I'm Daniel Kanyos, and as Vandana stated, I'm the Director for the Office of Clinical Evidence and Analysis in the Office of Product Evaluation and Quality within the Center for Devices and Radiological Health at FDA. I'll be providing CDRH's perspective on clinical evidence generation and discussing its uses for regulatory decision making and FDA's role in this wider stakeholder community and the importance of external evidence methodology in that. CDRH has been actively engaged in improving upon the clinical trial ecosystem for almost a decade. Recognizing the challenges with conducting clinical trials, we made strengthening the clinical trial enterprise a central strategic priority. We established a clinical trials program and a director, which is now rolled into our office of OSEA, and worked with the multi-stakeholder community on programs such as the External Evidence Methods Group and the establishment of a National Evaluation System for Health Technologies, or NEST, all aimed at more efficiently generating clinical evidence. CDRH has implemented processes and collaborated externally to streamline and support innovation as part of the clinical evidence generation through legislative policy, guidance activities, improving regulatory efficiency and predictability, developing the MDIC early feasibility study, clinical trial site network, collaboration on the in silico clinical trials, supporting real-world data collection and analyses, and awarding a collaborative agreement to establish that National Evaluation System for Health Technologies Coordinating Center. The MDIC Early Feasibility Study, or EFS working group, is a great collaboration amongst the EFS ecosystem of industry, investigators, and clinical sites. Through this, we have test-driven tools to increase efficiency of the EFS startup process. All the contributions from the FDA work to aim to achieve the CDRH vision that patients in the United States have access to high quality, safe, and effective medical devices of public health importance first in world. The in silico trials work leverages physics-based computational models and simulations of the device functioning within beating human hearts constructed to represent a virtual patient population. And the External Evidence Methodology Work Group, the External Evidence Methodology EEM, was established in 2019 to provide a more predictable pathway for use of external evidence methods. Innovative methodologies such as frequentist and Bayesian methods and cataloging the existence of these methods for evidence fusion from data external to a clinical trial. And the need for this flexibility in how we assess evidence is built into FDA regulations in describing what we may consider as valid scientific evidence to demonstrate a reasonable assurance of safety and effectiveness for marketing. Our regulations can consider many different types of studies and data as supportive. And so we have been well situated to accept real-world evidence for many years. Real-world data sources can include diagnostic lab and imaging data, unique device identification, patient and device-generated data, electronic health records, medical billing claims. So why use real-world evidence and real-world data in regulatory decision-making? Well, traditional clinical trials evaluate device performance in controlled settings. Benefits certainly are the control over the study design and protocol, control for confounding, but there are limitations, usually expensive and time-consuming, may be difficult to collect rare outcomes, and generalizability of the results can be a challenge. Some potential benefits of utilizing real-world data sources include a possible better understanding of device performance in the real-world environment, which can further inform the benefit-risk profile. Facilitation of collection of outcomes, not always feasible in traditional studies, such as longer-term outcomes, the ability to assess rare outcomes, performance in diverse patient populations, opportunities to partner with patients in new ways, collecting patient-reported outcomes, integrating mobile medical device apps, wearable devices, user experience, and ultimately reducing time and cost to answer important questions. The NEST Coordinating Center, NEST-CC, strong and growing network of collaborators enables access to a wide swath of evidence from clinical experience, which can be harnessed to support regulatory submissions. The network's institutions have access to robust data organized into standardized common data models. There are currently 14 network collaborators representing over 141 million lives. So the access to this real-world evidence is growing, increasing, especially with the development of the NEST-CC and the growing number of network collaborators within that. Further, NEST Coordinating Center in 2018 established a multi-stakeholder subcommittees to support efforts to conduct real-world evidence studies for medical devices. This resulted in NEST-CC publishing the methods framework, defining the key components of a study protocol for the evaluation of medical devices, and also the data quality framework, covering data governance, characteristics, capture, transformation, and curation. This was released in February of this year, really starting to illuminate that pathway for leveraging of real-world evidence. So starting with that access to this over 141 million records and providing the framework documents to really help illuminate the pathway for leveraging real-world evidence for regulatory decision-making, payers, and patients, et cetera. So with the full launch of NEST version 1.0, this large research network for real-world data and expert investigators is open to the medical device stakeholder community at large. This is an exciting time for real-world data, real-world evidence, and especially for leveraging that information, leveraging external evidence methodologies, and to really streamline the conduct of clinical trials and really arrive at that more timely answers to important questions. So having it start in 2019, the External Evidence Methods Group has an extremely bright future on the horizon. Thank you. I'm now going to pass it over to Dr. Ram Tiwari.
Hi, good afternoon, everyone. This is Ram Tiwari. I'm the director for the Division of Biostatistics in CDRH. I would like to thank Daniel for giving a good overview of RWD and also NEST-CC. I would also like to take this opportunity to thank Jitesh for coordinating the efforts on MDIC's External Evidence Methods Framework. The External Evidence Methods Framework is being developed under the umbrella of MDIC, and we have participants from industry as well as from FDA. MDIC External Evidence Methods Program was launched formally in April 2019 with a workshop that was held at the MDIC headquarters. Prior to that, there were two other workshops that were held, one at the FDA campus and the other at the MDIC center. In those two workshops, we covered Bayesian dynamic methods for borrowing, and after that, we decided that we need to expand the real world data to a broader term that is external evidence. And this word, external evidence, was coined by our center director, Jeff Suren. So the external evidence methods framework has three broad scopes. One is to categorize different sources of external data. The second one is catalog existing statistical methods for leveraging external data. And the third one, include examples to illustrate use of statistical methodology. We will also identify gaps in application and development of statistical methods. So let me begin with giving the definition of external data. This is very much similar to real world data. External data consists of the data that is collected from outside of the planned clinical study. Also, external evidence is clinical evidence generated by appropriate analysis of external data. The external data sources consists of the real world data sources, as well as it includes historical clinical studies, that is from OUS or US, as well as computational modeling and simulations. Also, it includes the laboratory test data. Potential use of external data include generating hypotheses to be tested in a prospectively planned clinical study, establish performance goal or objective performance criteria as a supplementary data, and provide additional information to construct investigational device arm or to construct a control arm or both. Finally, to augment an investigational device arm or a control arm, or to augment both investigational device arm and control arm. Statistical methods for generating external evidence include propensity score methods, Bayesian hierarchical methods, Bayesian commensurate prior method, Bayesian power prior methods, Bayesian dynamic power prior methods, propensity score integrated power prior methods, and propensity score integrated composite likelihood methods. These are just some of the methods. This is not an exhaustive list of all the methods. I would like to spend a few minutes here on the propensity score integrated approach. This method, as well as the two-stage propensity score design, has been developed by CDRH statisticians. Here, the propensity score integrated approach consists of using the propensity score as well as either power prior method or composite likelihood approach. If you look at the slide, there are three boxes here. The top box consists of the preliminary study design, where we define the primary endpoints and secondary endpoints, also determine the sample size, identify the baseline covariates, also identify the inclusion-exclusion criteria. At the second stage of the design, we use the propensity score to borrow subjects from external data sources. External data sources could be multiple RWDs, that is registries, or it could be historical control data or historical clinical trial data or OUS data. So we use the propensity score to match the subjects from the current study to one or multiple RWDs. And using this matching score, we come up or select the subjects from the external data sources. Generally, we propose that we use 5 quintiles to stratify the subjects and select the subjects from the current study and external data sources. We also consider a measure of similarity for each of the stratum, and using this measure of similarity, we come up with discounting parameter, which is used in the third stage, that is in the outcome analysis. So in this last box, in the outcome analysis, we derive the likelihood function for each of the stratum and obtain the parameter estimates using the discounting parameter. And then we combine the parameter estimates from each of these 5 quintiles and obtain an overall estimate. We can use the Bayesian power prior method, or we can use the frequentist composite likelihood estimate to estimate the parameter, a point estimate, or obtain the posterior distribution of the parameter. Pre-market regulatory considerations are important. Evidence derived from the external data can be used for approval or clearance of a new device. Also, it can be used for indication expansion for device that is already on the market. Important considerations. In using the external evidence, we need to follow that the external evidence is fit for purpose. The external evidence or RWD or external data source is relevant, reliable, prospectively designed clinical study. Also, we need to ensure steps are taken to alleviate bias. Other clinical and regulatory considerations are also needed. For example, we need to address the missing data, and we may need to also consider doing the sensitivity analysis to alleviate the bias. MDIC EEM survey that was conducted in June 2020 has the following objectives. We want information on identifying examples of use of external data in approved or cleared medical devices. We would also like to have information on extent of use of external data in ongoing clinical studies. Thank you. I would now turn it over to Ted Listick, who will cover the survey results and will have some final words. Thank you very much again.
Thank you very much, Ram. I'm Ted Listick. I'm the Senior Director of Corporate Biostatistics at Medtronic, and I'm very happy to join my other esteemed colleagues on this presentation today, and looking forward as well to our forthcoming live panel discussion with those of you out in the audience. So today I'm going to talk a bit more about some of the examples that we became aware of through the survey, as well as some other related applications from the external evidence, and talk a little bit as well about the methods aspect, not as in depth as Ram did, but more in the context of these various examples. So I think it bears reiterating this concept of what we mean by external data. So external data is data that's generated outside of a contemporaneous clinical study. And as Ram indicated, this includes historical clinical trial data, modeling and simulation data, registries, electronic health records, or administrative data, such as claims, billing, and records. Those last two items are examples of real-world data, but this is to underscore again that we think that external data is something that is greater than real-world data. Real-world data is certainly an important component, but not the only component. When we're talking about external data, we're usually considering the combination of some external information with some currently generating clinical study information. And actually, if you look on the picture on the right, I'm deliberately showing a picture of two streams coming together, one with a lot more suspended particulates than the other, the different colors. And so you could conceive of the idea that you have two streams of information, some with different other things inside it than the others, and you're looking to mix those two things together, one that you start with and one that's coming in external. So again, we're looking conceptually for the combination of some external information with some currently generating clinical study information, at least for this working group. And for right now, I'm going to focus on those aspects where we've got that mixture. I'll allude to something where it's external only elsewhere as well. So one way to think about this for the concept of external data is to think sort of a pictogram representation of the types of information that we could consider in a clinical trial. So it's a busy slide here, but I put lots of different icons in here to help drive your thought processes. So think of the blue large people as the information from one particular arm in a clinical study, and the yellow would be the control arm, And the large ones correspond to the ones in an ongoing trial. The smaller outlines of people on the left, that would correspond to data coming from a historical trial, right? So you can have your current information, you can have your historical data. For the current patients, you might also have information around their electronic health records, the thing that looks like a small laptop, information around the billing. You could have information coming from something like an Apple Watch. You could have information perhaps from a PRO that they filled in on their smart form. You might have information that's coming from a smart device, that it's downloading information about what actual steps the device is taking or measurements that it's recording. In addition to all this, you could even have modeling and simulation, that icon on the left-hand side of the page with the 5-pointed star and the 2 pieces of paper. So these are all different types of information, data sources that could potentially be pulled in aggregate to make an overall assessment of a particular device being studied. So one example here is something that I get to talk about a bit more. We were submitted as part of the survey, but it's something I know about because it's coming from within Medtronic. So this is a product of ours, the Impact Admiral drug-coated balloon, that's intended to treat peripheral artery disease of the superficial femoral or popliteal arteries. It has multiple different diameters, multiple different lengths. The drug being used is paclitaxel, and the excipient is urea, which facilitates drug transfer, is naturally occurring and non-toxic. So the Admiral drug-coated balloon was a Class 3 combination product. The PMA was initially approved on December 30th, 2014. And we made a couple different indication changes to it over time. And the part I'm going to focus on here is highlight in yellow, the second indication change, where we added in in-stent restenosis. And so you're seeing on the bottom in the highlight text sort of where we end up after all those changes have been made. So in-stent restenosis in PAD is an unmet clinical need where the presence of a foreign body, the stent, stimulates proliferation of the smooth muscle cells, resulting in the neointimal hyperplasia and the re-narrowing of the artery. So the management of ISR often requires repeated and complex interventions, which can impact both quality of life and healthcare costs. So this was a significant unmet clinical need, as the current approved indication prior to this included native vessel treatment only, did not include dealing with the subsequent inflammation, and the existing approved treatments had low effectiveness results. So in our overall Impact SFA Global Clinical Program, we had a US and European study, the Impact SFA 1 and 2 trial with 331 subjects. We had the Impact Global study with over 1,400 subjects. We had an Impact SFA 2 PK sub-study with 24. We also had a study in Japan and a study within China. And we had 3 different pre-specified imaging subgroups. Well, in order to go after indication expansion, we made an ISR PMA cohort with some subset of patients in the Impact Global study meeting the inclusion-exclusion criteria, which was in size the total of 164 subjects. So the strategy here was that we approached the FDA to discuss utilizing the Impact Global Registry for the ISR subjects. The FDA required a control arm to assist with the risk-benefit ratio and labeling. Sometimes you might go after this from purely characterization. In this case, you also want to do a comparison. FDA and Medtronic discussed the use of a national registry to obtain data on a control arm for the PTA subjects. And they agreed that the Vascular Quality Initiative, or VQI registry database, could be utilized for these subjects. The pre-specified plan how to analyze the data was agreed to, and Medtronic utilized a third party to analyze the data. There's multiple discussions with the FDA utilizing the pre-submission process. And the final plan, which the highlight of which I'm pointing out here in the yellow text with the purple arrow, the clinical evidence is that an observational propensity score adjusted comparative analysis of the Impact Admiral DCB from the Impact Global study against the PTA subjects from the Society of Vascular Surgeons VQI registry database. Right, so there you're starting with the global study subjects and bringing in as external subjects that had been in the VQI registry database. This general approach is actually the most common form of studies using external evidence that we've seen reported in the survey. The general approach is that within your study, you have just that one arm of subjects, here like the blue person icons, the large ones. And then external to that, coming in from a different source, you have these small yellow icons on the side. So this is coming from registry. So you're studying your one arm, but it's not enough to report on that one arm. You want to make a correct comparison, analytically sound, with another group. And so you pull in data from another source. So this is not, the concept of pulling in subjects for this is not novel. It's been done. But what we're doing now is viewing this particular example as something within a larger setting of multiple ways in which you could pull in information from external evidence and have an overall framework to think about that. So another example is something that came from a work group that preceded the external evidence methods working group. And so this had been a partnership where Medtronic employees and FDA and other industry persons worked together with MDIC to collaborate on a successful effort to develop a new statistical methodology for incorporating prior information in clinical trial analyses. This was a variant of some existing Bayesian analyses using a power prior. The main paper focused on using engineering models for prior information. The current work extends that to using clinical trial information where the applicability of the historical data is in question. We had a great workshop in 2018 prior to, again, this EEM getting going in 2019. And at that time on the campus, we had strong endorsement from both Prem Zuckerman and Ram Tiwari, a prior speaker, in the presentation today. So this is something, again, that is a subset of what we're doing and leads into the general work we're interested in within this work group within MDIC. So right now, we've got at Medtronic a clinical study going on with historical data. Actually, it's the earlier part of a pivotal study. We had some subjects. You see them indicated with the icons on the left-hand side. They're the small ones from our spiral HTN-off study, where we've examined the results, and the results have been published during the negotiations for our pivotal study. In the pivotal study, we are using patients from the early part of the spiral HTN-off study to contribute to the full analysis of the pivotal study. And this leads to an overall lower sample size than running two independent studies. So it's something where, naturally we had to develop the appropriate methods, and there's some discussion back and forth in terms of, what are the key elements you're looking to have as most important in your analysis plan. And as it's a new method, we're also having quite a number of discussions around using various sensitivity analyses to determine whether the assumptions we put in place are sufficient and how robust the findings are going to be as well. But again, this is an example where you're in fact pulling in information from both the control and investigative arm from prior information with current information on the control and investigative in the ongoing study. So another example is one where we have not yet gotten to a regulatory decision, but I'm pulling this up. We started with one where the decision has been made, another one's ongoing, and this is one where we are planning. So this is virtual patients in a planned pivotal trial for diabetes. So key stakeholders within our diabetes group expressed interest in exploring some alternative study designs, and they know that in the near future, they're going to have a series of product improvements, both on the algorithm side, variations on a closed loop, think about the artificial pancreas you may have heard of, as well as improvements with sensors that are better at determining and processing the information around blood glucose. So this presents an opportunity to think more about a program of trials rather than just focusing on individual trials in isolation. And we already have a published virtual patient model for glucose dynamics that informed some of our decisions about how we would perform the insulin dosage as a function of the measured glucose values. So this is an example, going back to that very first slide where I had all the different types, where you see up on the left that little box that's supposed to indicate the modeling and simulation. So here, we will enroll subjects in the study, the plan. We'd have the control and your active group. But we have decided to pursue a virtual patient strategy for diabetes, where the current virtual patient is a metabolic model that predicts glucose response to different insulin therapy regimes. And we have a very large pool, thousands of virtual patients developed from real data. So this opens up the possibility of providing virtual patients that are closely matched to the real patients we encounter during enrollment, or perhaps to provide virtual patients in subgroups that are otherwise very hard to enroll. So you get a better sense on as a whole within not just the enrolled population, but within the target population, what we believe that the likely response to therapy will be. So then briefly on methods, going back to some of the things that Ram talked about earlier. So there's a variety of methods that have previously been used to support regulatory decisions based on external evidence. There's a long history of working for the creation of performance goals or objective performance criteria. There's a number of different applications of propensity score matching or weighting approaches. We've seen some submitted decisions that come out through using Bayesian hierarchical modeling or a Bayesian commensurate prior approach. There are some additional newer methods that have potential for further regulatory decision making. So cardinality matching is something that's coming up. It's quite interesting, which gives us a different way of thinking about this as opposed to propensity scores. You can think of it broadly as somewhat analogous to the difference between randomization versus balancing, right? Now, how much are you concerned with what happens patient to patient versus having good balancing across the arms? There's also the discount power approach, which has been not used a ton, but it's one of the examples I talked about with the spiral study. And certainly the two methods that Ram alluded to, propensity score integrated approaches that are being developed between FDA and Johns Hopkins, those also have a lot of promise. Looking forward to seeing them continue to be developed. Overall, the results from our survey, it's very useful in an inclusive documentation sense. So we can match responses against public regulatory decisions. You know, people tell us they did certain things. We can look at the number, pull it up from the database and find out, yep, okay, that was that particular method which was described. But this survey is not an exhaustive census. There's likely many intriguing methods and examples that were not reported, right? Because we asked people to say, tell us things that you have a decision for. Please share what you can in terms of things you're planning. But we know there's other things out there that perhaps have not been shared broadly. But we're hoping that a number of people have shared relevant information, and this can allow us to understand better as a community what's being effective, what should be considered, how to move forward. So in conclusion, the concept of working with multiple sources of information is not new. There's many established methods out there, but there's still room for new methods development or application of unusual existing approaches. For a lot of sponsors, there's challenges in leveraging external evidence, and they can be more related to non-methods questions, identifying your potential data sources in the first place, establishing the relevance and reliability, and gaining alignment that the proposal you have is appropriate for a particular regulatory decision. A given data set might be perfectly adequate for doing signal detection, but not of a high enough quality that be needed to do indication expansion. You know, these are discussions that need to be undertaken with the agency. As a community, we will make progress when we can share pre-competitively lessons learned and make improvements to the process of using external information. So with that, thank you very much for listening to our early version of this, and we look forward to conversations with you now in the Q&A section.
Thank you for attending MDIC's annual public forum. We hope you will join us for next week's session, where we will discuss how to apply MDIC's newly released framework on real-world evidence generation for in vitro diagnostics and an update on MDIC's COVID-19 Real-World Evidence Project. Visit apf.mdic.org for more information and to catch up on previous sessions.